students. Today we're going to talk about general functions of sensory receptors. So sensory receptors are things that pick up information about both the internal and external environments of the body. They respond to some kind of stimulus, which is some kind of force or energy that's applied to the receptor, and then they need to transduce that force or energy, that stimulus, into electrical signals because that's what our nervous system runs on. Transducers are properly known as receptors, and they're going to convey signals to the central nervous system using sensory neurons. Now, there's a receptive field that allows the sensory neurons to pick up their specific information. A smaller receptive field allows for more precise stimulus localization, whereas a large receptive field gives us a really poor localization of the stimulus. For example, if you take a pen and poke your finger, it's really easy to tell exactly which part of the tip of your finger is being poked. On the other hand, if somebody pokes you in the back with a stick, it's really kind of hard to localize exactly where that's at, down to even a couple inches sometimes. It's hard to tell. That's because the fingertips and the face and the lips have very small receptive fields, and things like the back and the legs have very large receptive fields, at least in the cutaneous sensation. So a sensation is a stimulus that we're actually conscious of. We pick up stimuli all the time, but we're really actually not very aware of most of it, and therefore it's not really a sensation as such. It's still a stimulus, the brain still registers it, but we're not consciously aware of it. So that sensory input, if it's going to go to the cerebral cortex as a stimulus, and we can perceive that, most of that information is actually going to go to different areas of the brain. For example, we're picking up information on what blood pressure is in all of our arteries. We're picking up information on how much salt and water we have in our blood. We're picking up information about what the pH of our blood is like, how much carbon dioxide is in our blood, what the acidity of our stomach is like. All of these things are stimuli, but they're not sensations. So a modality is the type of stimulus that a particular sensory receptor is able to transduce. For example, your eyes. Your eyes have specialized phototransducer cells in the back of your retinas, and those phototransducer cells are able to take light energy and transduce it into electrical energy. Nowhere else in the body do you have photoreceptors, so if we shine light onto the back of your foot, you still can't perceive that. The location of a given stimulus is determined by the receptive field that it's picked up on. Those areas, those receptive fields, are directly mapped to an area of the brain called the postcentral gyrus. So that postcentral gyrus has a kind of little distorted map of the body called a homunculus, and we use it to represent just how much sensation is coming from any given part of the body. Now remember, with sensory receptors, with the action potential, as we depolarize and then we come back down and repolarize, moving negative or away from zero, we have that undershoot, that little area where we're going to hyperpolarize for just a few milliseconds. And that hyperpolarization means that it's more difficult to fire that same sensory receptor during that period of time, and it's going to require a stronger stimulus. So stronger stimuli are going to be able to cause that neuron to depolarize again within that time frame, and therefore it can depolarize at a higher frequency. The brain interprets that as a stronger stimulus or a more intense sensation. Now, one of the cool things about this whole system is that we've got receptor adaptation, and this helps to determine just how strong a sensation is at any given time. The best way I've found to describe this is using the zero or the tare function on a scale. For example, when I bake bread, I do it by weight. So I put a bowl on the scale, and then I push the zero button, and the scale now is ignoring the weight of the bowl, and any flour I add to it comes up on the display, so I don't need to do any math. So some of our receptors will adapt very rapidly. They will find a new zero, and we're no longer aware of any kind of stimulus. There's no more sensation. Other receptors, on the other hand, never really zero out. They will continue to tell us about that stimulus, so we'll continue to have a sensation for whatever that modality is. And it just honestly depends on the type of receptor. So tonic receptors really don't adapt. They're always going to be telling you what's going on. For example, the position of the head from the inner ear. All of your pain receptors are very tonic. They don't pick a new zero. Phasic receptors, on the other hand, are going to adapt rapidly. They only really respond to new stimuli, and if the stimuli isn't changing, they're going to go ahead and kind of reset themselves to a new zero, and you'll no longer be aware of that stimuli. So for example, pressure receptors or smell receptors. Maybe you've been holding hands with somebody while you're watching a movie. You completely lose that sensation of holding their hand as you become engrossed in the movie, and those pressure receptors reset themselves to zero. And then all of a sudden, as you or the other person move a little bit, then you've activated different pressure receptors. You've taken some of the pressure off some of the ones that you were previously activating, and all of a sudden things have changed, and now you're going to have that sensation. Smell receptors are the same way. If you've ever been in a fairly fragrant situation, wait about 15-20 minutes and you'll no longer be able to perceive whatever the smell is. As soon as you go outside for 20 minutes and come back in, you're going to start to smell it again, though. Here's a question. Why do you think pain receptors are tonic? I mean, wouldn't it be a good idea to, once the pain receptors let you know that there's been damage to the body, if it's stopped annoying you? Well, actually, no. The tonic receptors are there with our nociception, or our pain receptors, in order to keep reminding us that what we've done is damage the body, and it's a way to teach ourselves not to do it again. Now, when we look at our different types of receptors, we can classify them several different ways. One is by whether they're general or special receptors. So the general receptors are fairly simple. They're distributed throughout the entire body. We have somatic sensory receptors and visceral sensory receptors, and these are going to measure things like touch and pressure and distortion and proprioception of joints and muscles and pain and temperature. The special senses are the 5 senses that you are probably thinking of as senses. That's going to be smell and taste, sight, sound, and balance. We can also classify our receptors by whether they're picking up information from outside the body, which is an exteroreceptor, or from inside the body, which is an enteroreceptor. And then we have a third classification called the proprioceptor that detects the movements of the body and the limbs. The proprioceptors tend not to be phasic. Instead, they tend to be much more tonic. It's always important for your brain to always understand where your body's at in space. You don't want to put your hand above your head and then have your brain reset that to be the new zero. Now, we can classify receptors by the type of modality that they are receptive to. For example, there are chemoreceptors. These are going to detect chemicals that are dissolved in a fluid. So we can use these for both internal and the external environment. We can smell food, we can taste food, we can measure levels of oxygen in our blood, we can measure our pH, we can measure the acidity of the stomach. Thermoreceptors detect changes in temperature. That includes receptors in the skin and in the hypothalamus of the brain. We have photoreceptors that detect changes in light. That includes both intensity, color, and movement. And we find the photoreceptors are limited to the retina of the eye. Mechanoreceptors are all over the place. They're going to detect distortion of a cell membrane. These include touch, pressure, vibration, stretch. The pressure receptors are known as baroreceptors. Our proprioceptors fit into the mechanoreceptor category. Tactile receptors, even the receptors for balance in the inner ear, are a type of mechanoreceptor. Nociceptors are everybody's least favorite. They detect pain. Nociceptors are everybody's least favorite. They detect pain. And this is basically from damage to the body, any kind of tissue damage, whether it's chemical damage or thermal damage or mechanical damage to anywhere on the surface of the body, or the musculoskeletal system. That's going to be picked up by nociceptors and then sent to the brain. Here's an example of some tactile receptors. The endings can be encapsulated or unencapsulated. What that means is that if a neuron receptor, a sensory receptor, has a little gizmo on the end that lets it transduce a specific modality, then that's encapsulated. Unencapsulated neurons are just like they sound. The dendrite basically just stops in the middle of the tissue somewhere, and that's where it sits. So unencapsulated tactile receptors are important. We can use free nerve endings as simple tactile receptors. We can use them in mucous membranes. They're mainly for pain and temperature changes, but we can also use a few for light touch or pressure. We have unencapsulated receptors on our hair roots. We call these hair follicle receptors. These are located deeper in the dermis, and they're going to detect when a hair shaft wobbles. Continuing on with the unencapsulated tactile receptors, we have tactile discs that specialize in light touch. Moving on to the encapsulated receptors, we've got end or Krauss bulbs. These are sheathed in connective tissue, and they detect pressure and low frequency vibration. We have Pacinian or lamellated corpuscles. These are wrapped in neurolemocytes and connective tissue. These are located deeper in the dermis, in the hypodermis, and even in the walls of some organs. These detect deep pressure, coarse touch, and high frequency vibration. There are Ruffini corpuscles. These are also wrapped in connective tissue. They measure deep pressure and distortion of the skin. Meissner's corpuscles. These are intertwined nerve endings wrapped in modified neurolemocytes. We find these in the dermal papillae, and they're for discriminative light touch. Now, one idea here is referred pain, and that is when the brain gets confused as to the location of an incoming sensory signal. Because most of our sensation, remember sensation is different than stimulus, is originating from skin and muscle, when we do pick up a sensation from the visceral side of things, the brain kind of assumes that it's just happening from the somatic side. So the somatosensory cortex is really kind of unable to determine the true source of the referred pain. Some examples would be a heart attack. A lot of time people will have pain in the left arm or the left side of the neck, and obviously your heart is not in your arm, and that's because the sympathetic innervation of the heart and the innervation of the skin regions for the arm and for the neck all come from the T1 to T5 segments of the spinal cord. The kidneys and the ureters refer pain to the inferior abdomen, and visceral pain is often conveyed along sympathetic nerves, but occasionally on parasympathetic. Here is a map of referred pain. No, you do not need to memorize this, but these are just examples of how the referred pain works. We call these referred pain patterns. So for example, the heart can cause pain in the left chest, the medial side of the left arm, and the left neck, which they're not showing here for some reason. The ureters, as we mentioned earlier, kind of come down and hit the lower abdomen and especially the inguinal region. One last thing here is phantom pain. This is sensation associated with a removed body part. If somebody's had a limb amputated, they can still experience pain in the limb that's no longer there. And that's because the pain is actually in the brain. So the pain can actually be really quite severe, and to treat it, one method is to actually use mirrors.